Okay, so our last speaker for today's symposium is Professor Jian Tang. He's an associate professor at Mila and at Montreal. He has done a lot of exciting work on drug discovery and protein design. He couldn't come in person today due to visa difficulty, so he will present on Zoom. So without further ado, Jian, please take it away.
Okay, so thanks very much, Wengong, for the invitation and also the introduction. It's great pleasure to be here. Unfortunately, I cannot come in person due to visa issues. Hopefully, we can meet in person next time. So in today's talk, I'm gonna talk about geometric deep learning for protein design. I'm gonna quickly introduce the background of proteins. So as we know, proteins are the workhorses in living cells. So in our living cells, most of the functions are actually carried out by proteins, right? And that's why understanding the functions of proteins, also designing proteins of new functions is going to be important in many different areas. For example, therapeutics, antibody design, and also enzyme design in synthetic biology. Okay. And then, so nowadays, we actually see a huge opportunities for machine learning for protein design because a huge amount of data sets is generated in biometric domain. Like for example, we have like tens of billions protein sequence available. We also have like hundreds of thousands protein structure available in the PDB database. Okay, so a lot of different machine learning techniques have been developed for protein modeling. So these approaches are mainly divided into two categories. So the first category is basically sequence-based approach. So we just represent proteins as amino sequence, and then we can use models like language models, transformers to model amino acid sequence. So this is one type of approach. So another type of approach is, okay, we can represent proteins as 3D structures, right? And then we can use approaches like geometric deep learning approach to model 3D structures. Okay, so in our approach, in our group, so we have been taking the structure-centric approach to model 3D structures with geometric deep learning. Okay, so in particular, so we mainly try to address three research questions. The first research question is, okay, so once we are given a 3D protein structure, how do we represent these 3D protein structures, right? How can we learn features out of these 3D protein structures? So these are first research question. The second one is, okay, how can we, as we know, for most of the proteins, we don't know their 3D protein structures, right? How can we predict the 3D protein structures from their amino acid sequence? These are the second research question. Of course, our end goal is always we want to design new proteins with generative models. So I'm going to mainly focus on the first research question. Okay. So in this work, so what we do is we actually develop a new 3D protein geometric encoder to encode proteins into continuous repetitions. So our input is a 3D protein structure, and then given this 3D protein structure, we first construct a graph out of this 3D protein structure. So what kind of graph we are constructing here? So this is actually a residue graph. So every node here is a residue, okay? And then how do we define the edges between the residues? So we mainly make use of two kinds of information. So first, the structure information, and the second is sequence information. So for structure information, so essentially what we do here, we just construct the k-nearest neighbor graph from the spatial information of the protein structure to define the spatial relationship between the residues. So this is from the structure information. And then we also make use of the sequential orders between the residues in the protein sequence to define another type of like edges between the residues. Okay, but anyway, so here we will end up with a residue graph, and this graph is directed. And then the problem of learning protein structure translate to learning the repetition of the entire residue graph. Okay, so here we actually propose two type of graph neural networks to learn this repetition. The first approach is quite straightforward. So we just do massive passing between the residues. Okay, of course, when we do massive passing between the residues, since there are different type of edges between the residues, so we will also take the edge type into consideration when we do the massive passing. Okay, so once we do the massive passing, so we can have a repetition for all the different residues, and then we sum up all the residual repetitions to get the repetition of the entire graph or the entire protein structures. So this is the first type of like architecture we propose. And for the second type of approach, okay, we're actually not doing massive passing between the residues. So instead, so what we do here is we're actually doing massive passing between the edges. Okay, for example, we can do massive passing between neighboring edges 2, 4, and 4, 7. Okay. And then when we do massive passing between the edges 2, 4, and 4, 7, so we also make use of the angle information between the edges into consideration. So by making use of the angle information, so we actually basically better able to make use of the 3D geometric structure of the proteins, right? And then once we do the edge massive passing, so every edge can have a repetition, and then we can aggregate all the edge repetition to get the repetition of the whole proteins. Okay, so that's another type of like algorithm, which we call like a GeoNet edge. Okay, and for both type of approaches, we actually train the model in a supervised fashion, which means we're going to train the neural network right from some labeled data in downstream task. So this is like supervised learning. Okay. And then, so we also pre-train the 3D geometric encoder from some unlabeled proteins. So here, the idea is very similar to what we do in image representation learning with like self-supervised learning. So the basic assumption here is, okay, we use contrast learning. We basically assume that, okay, for two structure motifs, if they are from the same proteins, so then their repetitions should be similar. Otherwise, their repetition should be dissimilar. So that's the high level idea. Okay. And then, so we evaluate the models on some downstream tasks, like, for example, enzyme commission, number predictions, or fold classification. These are some classical protein understanding tasks. And then in terms of pre-training data sets, so we actually try different pre-training protein structure database. One is PDV experimental structures. Another one is the predictive structures by AlphaFold 2. Okay, so here we can quickly look at the results of the supervised learning performance. So we compare our approaches with some like sequence-based and also graph-based. So our approach is able to outperform both the sequence and the graph-based approach, which is not surprising because we make use of the 3D structures, right? And then in particular, if you compare the GeoNet and also the GeoNet edge. So GeoNet only makes use of the node which do massive passing between the residues. But for GeoNet edge, we actually make use of we do massive passing between the edges, which is basically better. Able to better make use of the edge, the 3D geometric encoder. So you can see the performance is actually significantly improved compared to the GeoNet. And then, so after doing pre-training, you can see the performance is further improved compared to the supervised training. So you see pre-training is indeed able to improve the performance. Okay. And then, so we also do some aberration study on the pre-training. So we actually try different pre-training database, either the PDB database, which are experimental structures, and also the AlphaFold to predict the protein structures. So we actually see that our pre-training model is actually quite robust, either trained on the experimental structures and also maybe also the predictive protein structures by AlphaFold 2. Okay. So this is the first work. And then we apply this kind of like a structure pre-training and the fine-tuning idea to a real-world problem, which is antibody optimization. So as we know in practice, sometimes the antibodies, like for example, the antibodies obtained through animal immunizations. So we usually sometimes we need to do lead optimization, right? So we need to do some mutations to improve the binding affinity. So in particular for this problem, so what we have is, okay, we have an antibody binding to a specific antigen, right? And then we want to make some mutations in the interface to improve the binding affinity. So mathematically, what we want to do is we actually want to build a binding affinity change predicted models. So we want to predict, okay, if we make some mutations on the interface, so what's going to be the binding affinity change, right? So we want to build this delta G predicted model, so which is the difference between the binding free energy of the mutant and also and the binding free energy of the wild type. Okay, and so this is the goal. Okay, so for binding affinities, essentially most the binding affinity most of the time we mainly actually look at the interface between the two protein structure, two proteins, right? So you see for this model, also we are also trying to model the protein structures, but for this problem, so we want to model the interactions between two proteins instead of like a single protein. Okay. So here we also, so this is a new work we published in this year's like Nature Communications. So these are new, also, so this for this work we also adopt the pre-training and also fine-tuning framework. So we first pre-train the model on a huge number of like unlabeled protein-protein complex structure, so that the model is able to understand how different proteins interact. Okay, and then we fine-tune the model based on some experimental data to build this like binding affinity change predictive model, and then we can apply this model to real world scenarios. We can do in silico screening and then identify some mutants which could lead to a higher binding affinity. Okay, so this is the main idea. So we can start from the pre-training stage, as I just said. So for pre-training, so we're gonna pre-train the model on protein-protein complex structure, because our goal is trying to understand how different proteins interact. Okay, so that's why we pre-train the model on protein-protein complex structure, and then so we also use like contrast learning idea. Okay, so what we do here is, okay, we take a natural protein-protein complex structure. We mainly take the interface. So these are positive pair, right? So this is a natural interface. And then what we do is we also make some mutations on the interface. And this is basically negative pair, right? So we do some mutations on the interface. Okay, we mutate the residues. We change one residue to another residue. But then for the mutated residue, we also quickly update the structure with some efficient sidechain structure predictive model. For example, we can sample from the Rotamo library. We can also optimize with some physics-based approaches, right? But then now we have these two different structures, and now we want to distinguish whether this is a natural structure, a natural complex, natural interface, or mutated ones, right? So we use the graph neural networks or geometric deep learning to encode the two different 3D structures, right? And then we build a classifier to distinguish the two, right? So this is the pre-training stage. Okay. And then in the fine-tuning stage, as we just said, we just want to predict, build a predictive model to predict the binding affinity change. So here we actually fine-tune the model with some experimental data. So in the experiment data like a ScanPy, so we have a protein-protein complex structure, and then we are given some mutations, and people already have tested the binding affinity change for different mutations, right? So that's the data we use to fine-tune the model. So here we do a similar thing, right? So we have the Y-type protein-protein structures or antigen-antibody structures, right? And then we have some mutations. We take the two structure as input, and you can get the repetitions of the two complex structures. But at the end here, we are not trying to do binary classification, because here we are actually trying to predict the binding affinity change, which is delta G. Okay, and so this model we're going to fine tune this model based on the experimental delta G data. Okay, by doing this, so we are able to build a predictive model to predict the binding affinity change given any mutations on the interface. Okay. So once we have this model, okay, we're gonna do some like evaluations, in silico evaluations. So here we do cross validations on the ScanPy data sets. So we compare with some traditional physics-based methods like FoldX, FlexCDG, and also some recent deep learning-based methods. So you can see our gigabyte can achieve better performance compared to traditional approaches. And then after doing pre-training, so our model actually achieved the best performance among all these methods. So this is some in silico evaluation. Okay. And then you may wonder, okay, can this model be applied to real world like problems? Yes, so we actually collaborate with some biologists for like antibody optimization. So here's a case like we have, so our like collaborator has a SARS-CoV-2 antibody. Actually, this is not SARS-CoV-2. This is an antibody collected, actually harvested from SARS-CoV-1 patient. And then they want to optimize this antibody so that it can target against the Omicron variant. So they already have this like antibody and also SARS-CoV-2 antigen complex. And then we use our models to do some in silico screening. So in the first round, so we identify some single mutations. We pretty much identify like, I think 7 or 8 single mutations and also the best double mutations. Okay. And then we test them in the wet lab. Okay. And then once we finish the first round of wet lab experiments, okay, we can identify a few good single mutations, And then in the second round, we just do combinatorial mutations. We mainly test like double mutations and also triple mutations. So at the end, we are able to identify a good mutant with 3 mutations. And these mutants, actually, the binding affinity has improved 17-folds. Actually, this problem is quite challenging because for the original antibody, the binding affinity is already like 1 nanomole, and now we're able to improve it to like 0.06 nanomole. So this actually, the problem is quite challenging. So this is the first case study we did with our algorithm. Okay. And then we actually also collaborate with another group which has a similar problem. So they also have like a SARS-CoV-2 antibody, and then they want to optimize this antibody to neutralize against the new variant JN.1. So we did a similar thing. So in the first mutation, in the first round, we do in silico screening. So we identify some good single mutations, and in the second round, we combine them to do double or triple mutations. Eventually, so we actually identify a good mutant, which the binding affinity has improved over 300 folds compared to the wild type. Okay, and this paper now is actually in revision in PNAS. So this is the second case study. Okay. And then, so I probably do not have time to cover the details of the other work we have done. So for 3D protein structures and also de novo design, I'm going to quickly summarize some work we have done in these 2 directions. Okay. So for the first work, DivPack, so we mainly work on side chain structure predictions. So you guys probably all know like models like AlphaFold2 or ESMFold, right? But remember that for AlphaFold2 or ESMFold, so they mainly work on like predicting the backbone structures of proteins, right? So actually, the side chains predicted by AlphaFold2 and the ESMFold, so they are not accurate. So that's why in this particular work, so what we did is we actually proposed diffusion models to accurately predict the side chain structures of protein. So side chain structure prediction is very important, like for drug design, because for either for small molecule or proteins, right? So interactions between 2 molecules is actually mainly happened through the side chain interaction. So that's why side chain structure prediction is a very important problem. So what we did in this work is we actually propose a diffusion model to generate or predict the 3D structures of the side chains. So essentially just generate the 3D coordinates of the atoms. As you know, because for side chains, so their degree of freedom is not the 3D coordinates of the atoms. So it's actually mainly controlled by the 4 torsion angles. And that's why in this work, we actually didn't do diffusion in the Cartesian space. So instead, what we do is we actually did a diffusion. We propose a diffusion model in the torsion space. So that's the main idea of this work. So I'm not going to the details. Okay. And then for the second work, this is also related to protein structure prediction. As we know, like for AlphaFold2 and also ESMFold, they only predict a single structure for each protein sequence, but we know like each protein can take multiple conformations, right? And that's why here we actually try to predict multiple conformations for given an amino acid sequence, right? So the high level idea is we got some inspiration from traditional like molecular dynamic approaches, especially the approach called like simulated annealing. Okay, and then we train joint models, essential diffusion models, to generate multiple conformations given a single amino acid sequence. So I'm not going to details. So this paper is accepted in this year's ICLR. Okay, and so this is the second one. And then finally, for the diffusion models, and this is a very latest work we did. Like we just put it on archive. So for this problem, we want to, given a small molecule, we want to design a protein binding to this small molecule. Okay, so here we actually propose a model to based on the flow matching framework to model the joint distribution of the protein ligand complex, and then given the chemical graph, so we can use flow matching to simultaneously generate the protein ligand 3D complex structure. Okay, so that we can eventually get like proteins binding to the 3D molecule, binding to the given ligand. Okay, so I will wrap here. So essentially, in our group, so we have been taking a structure-centric approach to model 3D structures of proteins. So we mainly make use techniques like geometric deep learning and also diffusion models, flow matching to model different kinds of problems in proteins. In particular, how to represent 3D protein structures, how to better predict protein structures like side chains, the dynamics, and also protein-protein complex structure prediction. Of course, at the end, we care about how can we design new proteins with diffusion models, with with models like flow matching. So that's all I want to show. Thank you very much. Okay.
Great. Thank you, Jian, for your wonderful talk. Any questions? I actually have questions about your design part, which you couldn't really go into too much detail. But how are you generating both the ligand and protein simultaneously, or you're given the ligand conformation is actually given?
Oh, that's a good question. So I think so for many of our recent work, the 3D ligand structure is given. So in all this latest work, we actually only give the 2D chemical graph as input. So the 3D conformation of the ligand is not given. Okay.
And do you know the binding pocket location, or you're just
no.
Okay.
We don't assume the binding pocket is given. The input is just arbitrary, so just a like ligand. So only the sequence or the chemical graph is given, and then we want to generate a protein which is able to bind to the ligand. But you're right. So we are also now doing some testing or proposing some new variants. So okay, maybe we know that we are the binding positions of the ligand. Okay, so in that case, we can also do some more controllable protein design. Okay, you want to design proteins binding to specific regions of the small molecule. Yeah.
Great. So since we are already over time, so let's give our speaker one more round of applause.
Thank you very much.